Good day, students. Welcome to zoology class. Hope all of you are fine. Today, in continuation to my presentations, today I will take up my next topic, and that is enzymes. So, today's presentation is on the topic enzymes. So, what are enzymes? So, in living organisms, chemical reactions are brought about by special protein molecules called enzymes. And these are the biological catalysts. Chemical reactions take place only when the molecules collide. And the energy which is required to produce the collision, it is known as the activation energy, and the high temperature provides the activation energy, but enzymes also provide energy and lower the amount of activation energy. An enzyme, a substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions proceed without itself being altered in the process. So basically, you know that many chemical reactions take place in our body, and all these chemical reactions, they are brought about by special protein molecules. Actually, the chemical reactions are catalyzed. It means their rate is increased because of certain special protein molecules, and those protein molecules are known as the enzymes. So enzymes are the biological catalyst. Whenever there is a chemical reaction, collision takes place. And when collision of reactants takes place, then energy is required. And that energy is known as activation energy. And the high temperature provides that activation energy for the reaction. But when there is present an enzyme or the biological catalyst, then enzymes also provide the energy. And when enzymes provide the energy for the collision in a chemical reaction, then the energy from the temperature or the heat is required in less quantity. So the activation energy amount is lowered. So we can say that the enzymes lower the amount of activation energy. So enzymes act as the catalyst in reactions going on in living organisms, and they increase the rate of chemical reaction without itself being changed in the process, and they remain same at the end of the reaction. So this is enzyme. Then what are the characteristics of enzymes? So as I told you that enzymes speed up the reaction by lowering the activation energy of the reaction, and their presence does not affect the nature and properties of the end product. So if enzyme is present during the chemical reaction, the end product will be the same. So there is no effect on the nature and properties of the end product. And these enzymes, they are highly specific in their action. That is, each enzyme can catalyze one kind of substrate in a reaction, and only small amount of enzymes is required for the chemical reactions. So enzymes are sensitive to change in pH, temperature, and substrate concentration. And there is a term turnover number. So what is turnover number? Turnover number is defined as the number of substrate molecules transformed per minute by one enzyme molecule. An example is catalase turnover number. What is the turnover number of catalase? It is 6 multiplied by 106 per minute. It means what is turnover? Turnover number is the number of substrate molecules which are transformed per minute by one enzyme molecule. So these are the characteristics of enzymes. So now you know that what are the characteristics of the enzymes. Next comes the classification of the enzymes. So enzymes are assigned names based on the one who discovered it. in the olden times. It means earlier enzymes were assigned names based on the discoverer, based on the name of the discoverer. And now with further researches, classification became more comprehensive. And now, according to International Union of Biochemists, that is IUB, enzymes are divided into 6 functional classes. And now the classes are classified based on the type of reaction in which they are used to catalyze, in which they are used. And those 6 kinds of enzymes are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So these are the 6 types of enzymes, or 6 classes of enzymes. Now we will talk about them one by one. So first is the oxidoreductases. What are oxidoreductases? These enzymes catalyze oxidation and reduction reactions. For example, pyruvate dehydrogenase. Then comes the this pyruvate dehydrogenase. It catalyzes the oxidation of pyruvate to acetyl coenzyme A. And these enzymes, that is the oxidoreductases, ये catalyze oxidoreduction between two substrates. And the next class of enzyme is transferases. And what are transferases? These are the enzymes. They catalyze the transfer of a group between a pair of substrate molecules. Then comes the hydrolases. Hydrolases are the enzymes which break a complex molecule into two products by breaking the bond. So these are the hydrolases. For example, lipase and amylase. These are the hydrolases. They break the complex molecule by hydrolysis, that is the addition of the water. So these are the first 3 classes of the enzymes. Next class of enzyme is the lyases. These enzymes break without the anything, and these enzymes break covalent bonds and remove groups without hydrolysis, and these catalyze the breakage of the bonds without catalysis. So this is the example, and these are the lyases. Then next comes the isomerases. Isomerases enzymes catalyze rearrangement of molecular structure. So this is isomerase. And the last is the ligases. These enzymes, they join together two molecules by synthesis of new covalent bonds. Means ligases catalyze the joining of the two molecules. For example, DNA ligase catalyzes the joining of two fragments of DNA by forming a phosphodiester bond. So these are the 6 types of the classes of the enzymes. So this is actually the classification of the enzyme. Now the next concept is of cofactors. Cofactors are non-proteinous substances which are associated with the enzymes. As you know that enzymes are basically proteinaceous in nature, and they have certain non-proteinous part, and that non-proteinous part is known as the cofactor, and cofactor is essential for the functioning of an enzyme, and an enzyme without a cofactor is known as apoenzyme. An enzyme and cofactor together constitute the holoenzyme. It means cofactor plus apoenzymes, they form the holoenzyme. And these enzymes, as you know, that they remain unchanged at the end of the reaction and can be used again, and they are extremely sensitive to pH, and each enzyme has its own range of pH, temperature, etc. to work efficiently. And now if we talk about the cofactors, there are three kinds of cofactors present in the enzymes. Number one is prosthetic groups. These are the cofactors which are tightly bound to the enzyme at all times, and its example is FAD. And next type of cofactor is the coenzyme. So coenzyme binds to the enzyme only during catalysis, and at other times it is detached from the enzyme, and its example is NAD. And next cofactor is metal ions, and for the catalysis of certain enzymes a metal ion is required at the active site to form the coordinate bonds, and its example is zinc. So this is the concept of the cofactors. Then next comes the naming and classification of enzymes. So the International Union of Biochemistry and Molecular Biology is entrusted with designating names to enzymes in addition to assigning a number in order to identify them. So the number of the enzyme is also given and name is also provided to each enzyme, and this work is done by the International Union of Biochemistry and Molecular Biology. And apart from the few originally studied enzymes such as rennin, pepsin and trypsin, almost all the enzymes names end in ase, and as per the standards, focal points of nomenclature of enzymes are both the type of the reaction catalyzed and the substrate acted upon. So earlier, the enzymes like rennin, pepsin and trypsin, they were named differently. But now the enzymes, they end with the word ASE. And now the focus point while naming the enzyme is the type of the reaction they catalyze and the substrate they act upon. So these are the two factors while naming the enzymes. And most commonly, enzymes are named to provide data on the function as opposed to the structure of the enzyme. So while naming, we don't see the structure of the enzyme, but the focus is on the providing data on the function of the enzymes. However, there are three significant features of nomenclature process of enzymes, and they are suffix. Suffix means the word which is at the end, and suffix ase is provided to the enzyme which recognizes the substance that it is the enzyme, and suffix in, like in rennin and pepsin and trypsin, this is observed in the name of the enzymes earlier. And now prefix is identified by the type of reaction the enzyme catalyzes. And for example, enzyme hydrolase, it catalyzes the hydrolysis reaction, and enzyme oxidase, it catalyzes the oxidation reaction. So how this is the naming of enzymes is done. Then in addition to the type of reaction, the identity of the substrate is also taken into consideration. For example, glucose oxidase. This is the catalysis of glucose oxidation. Then lactate dehydrogenase. This is the catalysis of eliminating hydrogen from the lactate ion. Sometimes the reaction type is given instead of substrate. For example, lactase. This is the hydrolysis of lactose is catalyzed. Then urease. In this, hydrolysis of urea is catalyzed. And I will give you one example of naming. As per the standard International Union of Biochemistry, name of enzyme comprises of two parts. Name of the substrate for the enzyme and type of reaction they catalyze. The second part therefore ends with ase suffix. For example, lactate dehydrogenase. So lactate is the substrate and dehydrogenase means it dehydrogenases, and at the end ase is written. So this is how the naming of the enzymes is done. And now next comes the factors affecting the enzyme activity. What are the factors which affect the enzyme activity? and the number one factor is the temperature. So, how temperature affects the enzyme activity? Enzyme activity is highest at optimum temperature. It is very inactive at low temperature, and at very high temperature enzyme gets destroyed, as enzymes are made up of proteins and proteins are denatured by excessive heat. For example, keeping milk in refrigerator prevents curdling because enzyme activity is stopped at low temperature. So, enzyme activity is highest at optimum temperature. At very low temperature, enzyme becomes inactive, and at a very high temperature, enzyme gets destroyed and there is no activity. So, this is the graph showing the activity of the enzyme depending upon the temperature. So, as the temperature rises, enzyme activity rises. And then at very high temperature, enzyme activity falls. So, this is the temperature. So, temperature affects the enzyme activity like this. So, this is the first factor, that how temperature affects the enzyme activity. Next comes the pH. pH is the second factor which is affecting the enzyme activity. Different enzymes have different pH, that is the optimum pH. For example, digestive enzymes work best in acidic pH, whereas intracellular enzymes work in neutral pH. So, this is how pH affects the enzyme activity. Next factor comes the enzyme concentration. Initially, as enzyme concentration increases, the rate of reaction also increases. And after a certain point, enzyme concentration is more than needed, so no enzyme substrate complex is formed, and therefore the rate of the reaction does not increase. So, this is the graph showing the effect of enzyme concentration on the enzyme activity. So, as the enzyme concentration increases, the rate of reaction increases, and the point comes then no more enzyme concentration increase is needed, so then the rate of the reaction does not increase. So, this is the factor of enzyme concentration, that how it affects. Next factor is the substrate concentration, and the behavior of the substrate concentration is the same as the enzyme concentration. That is, that as the substrate concentration increases, rate of reaction also increases, and then a point comes then no more substrate concentration is required to be increased, and then the rate of reaction does not increase. So, these are the factors which affect the enzyme activity. Now, next comes the inhibition of enzyme action. The activity of enzymes can be inhibited by compounds, and those compounds are called as the enzyme inhibitors. It can be reversible or irreversible. In some cases, the inhibitors can be removed easily from the enzyme, while in other, the inhibitor blocks the enzyme, causing a damage in its structure, making it non-functional. And these inhibitors or the inhibition of enzyme action can be studied under certain headings, and the number one is competitive inhibition. And what is competitive inhibition? Substances are the inhibitors which are structurally similar to the substrate and compete for the active site of the enzyme are known as the competitive inhibitors. And usually such inhibitors show a close structural resemblance to the substrates, to the enzyme they inhibit. In such a case, in spite of enzyme substrate complex, enzyme inhibitor complex is formed and enzyme activity is inhibited. It means enzyme reacts with inhibitor and then enzyme inhibitor complex is formed in place of enzyme substrate complex. So, this is the diagram which shows the competitive inhibition. So, this is the enzyme, and all enzymes, they have a active site here where the substrates get attached. And if the competitive inhibitor is present of the same shape, then what happens? This substrate does not bind with at the active site, and in this place competitive inhibitor, it binds at the place, and then enzyme inhibitor complex is formed in place of the enzyme substrate complex. Now, next comes the non-competitive inhibition, and these substances are the non-competitive inhibitors, do not combine with active sites but attach somewhere else and destroy the activity of the enzyme. Both enzyme inhibitor and enzyme substrate complexes are formed. An inhibitor binding alters the three-dimensional configuration of the enzyme and thus blocks the reaction. Non-competitive inhibitor do not compete directly with the substrate for binding to the enzyme in it, and the non-competitive inhibition cannot be reversed by increasing the concentration of the substrate, that it is irreversible. For example, cyanide inhibits the mitochondrial enzyme cytochrome oxidase, which is essential for cellular respiration, and this kills the animals. So this is the non-competitive inhibition. So more AMP is a non-competitive inhibitor of fructose biphosphate phosphatase, the enzyme that catalyzes the conversion of fructose 1,6 biphosphate to fructose 6 biphosphate. So this is an example of the non-competitive inhibitor. So this diagram shows the non-competitive inhibition, that this is the enzyme, this is the active site, and in addition to this active site, here the non-competitive inhibitor, it binds, and here substrate is binder. So it means that in this case both the enzyme inhibitor complex is formed and enzyme substrate complex is formed, and so the reaction or the enzyme activity, it is lowered. So this is a non-competitive inhibition. Next comes the feedback inhibition. In this type of inhibition, in number of cases, accumulation of final product of the reaction is capable of inhibiting the first step of reaction. So the enzyme E1, it catalyzes from A to B reaction, and enzyme 2 catalyzes the reaction from B to C, and in the same way E3 enzyme catalyzes the reaction from C to D, and E4 catalyzes the reaction from D to P, and then P is the final product, and when final product is formed, the accumulation of the final product, it inhibits the reaction, and it does not allow the formation of B from A. So the enzyme 1 activity is stopped. So this is feedback inhibition. So the product P checks the activity of the enzyme which converts A into B. It is quite useful mechanism because it checks the accumulation of products and the phenomena in which the end product of the metabolic pathway can regulate its own production by inhibition of the sort. This is called as feedback inhibition or negative feedback inhibition. This type of inhibition can be shown in Escherichia coli bacterium from a substrate threonine by a series of intermediate reactions. So this is the feedback inhibition. Now the last type of inhibition is the allosteric inhibition or the modulation. Allosteric literally means another place, and still other inhibitors join an enzyme at a specific site and change the form of the active site meant for the substrate. These inhibitors are known as modifiers or modulators, and the sites where they fit in are called allosteric sites, and modulators are of two types, positive and negative. Change of active site form prevent the binding of the substrate to the enzyme and stop the reaction. So this is the allosteric inhibition. So the process is called allostery or the allosteric inhibition, and the enzyme with allosteric sites are called allosteric enzymes. Jacob and Monod have termed the phenomena as allosteric transition. An example of allosteric enzyme inhibition is hexokinase that converts glucose to glucose 6-phosphate. Glucose 6-phosphate causes allosteric inhibition of hexokinase, and this is called feedback. So this is about the inhibition of enzyme action. So this is all about for enzymes. So today you have studied about what are enzymes, what is their classification, how they are named, and what is inhibition of the enzyme action. And so these are the certain concepts which you have studied today in the topic on enzymes. Now I have already sent you study material on enzymes. Now you have to go through this video presentation, then you have to prepare notes by combining the material of the presentation as well as the study material. And in case you find any difficulty, then you can come to me and discuss with me in the class. Thank you.